So, hi you all. Good evening, good morning, good afternoon. Welcome to the SCAR Open Science Conference 2022 edition. And especially welcome to our session, Environmental Factors Driving Diversity and Composition of Fossil and Living Antarctic Communities. I am Fernanda Quaglio from the Federal University of São Paulo, Brazil, and together with Fabiana Canini from the University of Tuscia, Italy. We are the convenors of the session with the contribution of Dr. María Eugenia Raffi from the Centro Austral de Investigaciones Científicas, Argentina, as co-convener. So each of you have 12 minutes to present your contribution. The question and answers will be after 4 presentations, so you can write your questions during the presentations in the question box. So we will read it during the question and answers time. We will have 2 times for questions in this first block of this session. The organizer sent us a message for all of us, informing all attendees that all the talks will be recorded and will be available in the SCAR virtual platform and also in the YouTube channel with 24 hours of the session. So if you wish to not publish your talk, please inform us to pause recording, okay? So I hope you enjoy your session and the discussion on the subject. And now I call Fabiana to present the authors and the works. Fabi.
Welcome everybody. Just to respect our schedule that is published, we will wait for the right time. So the first speaker for today is Beatriz Pinheiro with a presentation on the diversity and community structure of bacteria and archaea of the Antarctic Peninsula Southern Ocean. I would ask the IT team to play the video that they have now, if possible.
Good afternoon, everyone. My name is Beatriz Riedo. I am an oceanography undergraduate student from University of São Paulo, Brazil, and I represent my scientific initiation project entitled Diversity and Community Structure of Bacteria and Archaea of the Antarctic Peninsula Southern Ocean. This work was guided by Professor Camila Signori and Laísa Faria from University of São Paulo. This presentation contains current scenario and study proposal, the study area, environmental factors, DNA extraction and analysis, results and discussion, and the take-home message. Usually, I start with an introduction about the importance of Antarctica, but I think here everyone knows. So, as do we know, the Southern Ocean is a major component of global ocean circulation and biogeochemical cycles, contributing with about 25% of atmospheric CO2 sequestration. Marine microorganisms play an important role in the biogeochemical cycling of nutrients and is the base of food chain. They are responsible for the majority of the metabolic activity of the ocean. In a climate change scenario, projections for the sea surface temperature, even in their most optimistic scenario, can cause change at all tropical levels, putting at risk the marine ecosystem of polar regions. This change generates imbalances in the structure of microbial communities, which are influenced by physical and chemical factors, such as temperature, salinity, dissolved oxygen, and availability of nutrients, combined with ocean circulation. Based on this study aimed to investigate the diversity and composition of the Antarctic marine microbiome in the austral summer of 2014 and 2015, relating to temperature, salinity, nitrate, nitrite, and silicate from 5 meters to 2019 meters depth. The study area is the Antarctic Peninsula, which comprises the Gerlach Strait, Bellingshausen Sea, Bransfield Strait, and Weddell Sea. Studies report that the west side of the Antarctic Peninsula had greater variations in the surface temperature and salinity than the east side. However, each part of the Antarctic Peninsula has distinct and seasonal physicochemical characteristics, circulation, and primary productivity. The data analyzed here were obtained from scientific expeditions during February and March from 2014 and 2015, aboard the polar ship Admiral Maximiano, with annual monitoring stations and water collections from different pelagic zones, from the surface to bathypelagic. Temperature, salinity, and water samples were collected through a CTD rosette system coupled with Ninsky bottles. Nitrate, nitrite, silicate, and phosphate were determined using flow injection analysis. A protocol for low biomass samples developed and adapted was used for DNA extraction. The quantitative insight into microbial ecology software was used to analyze the DNA sequence. For statistical analysis and figures, software R, Excel, and Ocean Data View were used. About the distribution of samples, it seems that they don't have a clear distribution, but these lower dots represent the deep sea zones, so it makes more sense. The February samples dominates the highest temperature, and the March samples, which represents the end of the summer, had an average temperature between 1 and 0 degrees and low salinity. About the nutrients, it was possible to observe that surface waters had higher concentrations of ammonia, fluorescent, and nitrate. Deep waters had higher concentrations of nitrate, nitrite, and silicate. However, no significant correlation was found. About the relative abundance of microorganisms by region, sea ice, Bellingshausen, North Antarctic Peninsula, Bransfield, Weddell, and Gerlach showed high abundance of bacteroidetes and proteobacteria phyla. The archaea, represented by Thaumarchaeota in gray, are commonly found in deep waters, which explain their absence in sea ice region. Regarding family, high abundance of Oceanopyrilalis, Flaviobacterias, and SAR11 were found in all the regions. Our results corroborate what were found by other studies regarding the relative abundance. Signori et al. 2014, Gonzalez Rocha et al. 2017, and Cal et al. 2019 also described proteobacteria and bacteroidetes as the most abundant groups for the region. A principal component analysis, PCA, a multivariate ordination with the sampling distribution and the environmental gradient, showed that in quadrant 1 and 4, samples with high diversity index were found at high depths and more saline waters. In quadrant 2, we find the surface samples and high temperature and less saline waters. In quadrant 3, shows that samples with low diversity are found in low saline waters. So, that could explain the difference in microbial communities, contributing to the conclusion of other works that microbial communities vary significantly with water column stratification. The alpha diversity index, in general, also showed a significant positive correlation with depth and salinity and negative correlation with temperature. Past studies also observed a positive correlation between salinity and silicates with the distribution of bacterial community composition. So, the take-home message: the environmental factors that reflect and determine the structure of the microbial communities are influenced by vertical stratification and by water masses. the relative higher abundance of proteobacteria and bacteroidetes and Tomarchiota are in agreement with other studies in the region. In a climate change scenario, changes in the physicochemical conditions of seawater can cause imbalances in the functioning of the entire marine ecosystem. Therefore, it is necessary to understand the dynamics of microorganisms and the impact of these variations on the marine microbiome in order to predict responses at different trophic levels and avoid future catastrophic scenarios. So, I would like to thank the laboratory colleagues of the Microcean Lab, CNPq for the granting the Scientific Initiation Scholarship, and for you, thanks for watching. And if you have any questions, please send an email. Bye bye.
Thank you to Beatrice, even if she's not present. We have to wait some minutes for the next presentation, just to be in time with the schedule. She's not present, so we cannot ask her to add something. Okay, we can move to next speaker, who is Maria Cristina Casero, and the title of the presentation is Disclosing the Core Cyanobacterial Community Dominating Glacial Retreat Areas on the Antarctic Peninsula.
Thank you so much. Can everyone hear me? Okay.
Yes, we can hear you.
Thank you very much. So, hello everyone, my name is María Casero, and I'm here to present a work conducted by Pablo Almela, Ana Justela, and Antonio Quesada and myself about the cyanobacterial community dominating glacial retreat areas on the Antarctic Peninsula. First of all, the climate change scenario has warmed the entire globe, and if it can have signs in every ecosystem, almost every ecosystem, the most visible signs are melting glaciers and ice caps. Actually, in the Antarctic Peninsula, in the last half century, the temperature rise 2.5 Celsius degrees, what lead to the loss of more than 28,000 square kilometers of the ice shelf, and also the recede or retreat of more than 87% of glaciers on this area. This glacier retreat is due to the loss of the ice mass balance, that is due to the less accumulation of snowfall combined with more ablation, what leads to a backward movement of the glacier front. This backward movement, this glacial retreat, gives new soils that can be exposed to the conditions. So it arises spatiotemporal gradient for the ecosystem development, what we know as chrono sequence, where the higher distance from the ice shelf would be a longer exposure time of the glacier forefield. These chrono sequences and this correlation between distance and time of exposure is really useful to understand and to study the development of soils. In recently deglaciated areas, microorganisms are the pioneer organisms that colonize these new exposed soils. And they're really a key organisms for the soil development and the primary succession because of their implication in the biogeochemical cycling. Among them, cyanobacteria, which are also the main primary producers and the Antarctic Peninsula, due to the ability they have to cope with polar extreme conditions, they have an important role in early succession stages because they can incorporate to the soils carbon and nitrogen. Previous studies says that especially non-stellar cystus filamentous cyanobacteria from polar and alpine phylotypes are the main pioneers in these communities. Thus, glacial retreat areas constitute an ideal scenario for investigating changes in the composition and structure of the microbial community over time. In this scenario, our main goals are to understand the primary succession of cyanobacterial communities in glacier forefields from Antarctica and to investigate the presence of a core cyanobacterial community, which would highlight the role of some groups in the Antarctic ecosystem. For this, 3 glacier retreat locations along the Antarctic Peninsula were studied. All of them were ice-free during the summer, vegetation-free, and of course, free of megafauna disturbances. 2 of them were located on Livingstone Island, Clark Nunatak, and Elephant Point, and both were limited with Roche-domed glacier. The third one was located in Amber Island and is called Biscoy Point. The 3 of them were ice-free for more than 50 years. Additionally, we decided to sample 2 cyanobacterial-based mats in order to understand if there's this core cyanobacterial community in the Antarctic Peninsula. And we took a sample in Byers Peninsula and another one in Horseshoe Island. How was the sampling? For the soils on the glacier retreat areas, in the 3 retreat areas, 3 sampling points were taken: NS1, NS2, NS3, ES1, ES2, ES3, BS1, BS2, with BS3, where 1 means the shorter distance from the glacier front, and 3 means the longer distance from the glacier terminus. So that 1 means younger soils and 3 means older soils. Also, microbial mats were sampled in Byers Peninsula from flooding area and in Horseshoe Island from stream. The estimation of the glacier retreat was done comparing Google Earth satellite images from 1990 to 2020. And of course, we did a chemistry analysis of the soils. To perform the molecular study, we did a DNA isolation followed by PCR. 16 general primers were used for a first step study, and then a specific cyanobacterial 16S primers were used. The sequencing was performed with Illumina MySeq, and also what we obtained were at the end amplicon sequence variants, ASVs. These were our results. Regarding the glacier retreat, what we could see with the comparison of the images is that the younger soils had between 5 and 20 years old, while the older soils had more than 30 years. So at some point, the intermediate soils, NS2, ES2, and BS2, had between 20 and 30 years old. In the soil chemistry analysis, even if we saw some trends in some of the parameters, none of them were significant. So that suggests that maybe these soils are not very developed primarily, and also that probably these soil characteristics or features are not really shaping what is going to happen in the diversity analysis that I'm going to show you. When we did the 16S general primers study, what we saw, first of all, is that in the 3 retreat areas, proteobacteria and actinobacteria were the main components of these communities, reaching between 66 and 72% of the relative abundance of the communities. If we have a look to cyanobacteria, that are those in black, what we see is that they differ not only between retreat areas, but also in soil ages in each retreat areas. What we saw in Biscoy Point, this higher relative abundance in younger soils and lower in older soils, is talking us about the cyanobacteria as pioneering organisms. So they are more important at the beginning of the development of the soil and then less important. But what we saw in Clark Nunatak and Elephant Point is possibly, or it suggests the presence of ancient or alloctonous organic matter that would limit the advantage these organisms have to not depend on extra nutrients. When we have a look using these 16S primers specific for cyanobacteria, what we see is that 2 orders are the main components of all these communities, oscillatoreals and pseudonavinails. What we see is that Clark Nunatak and Elephant Point have the same shift, where oscillatoreals are dominant in younger and older soils, while pseudonavinails are dominant in intermediate soils. Then, when we have a look in Biscoy Point, what we see is that oscillatoreals are the dominant in all 3 soil ages, and it looks pretty similar to what we see in the microbial mats. Then we go deeper and have a look into genus level. And what we see, first of all, that these pseudonavinails are mainly composed by one single genus, Pseudonavinae, especially in Clark Nunatak and Elephant Point. Then when we have a look to the oscillatoreals, it's a mix between Microcoleus, Formidasmis, and Formidium in the glacial retreat areas. It's not the same when we have a look to the microbial mats, when Gatelerinema and Ticonema are the main genera found in these samples. In any case, all those are cyanobacterial, non-heterocystous filamentous cyanobacteria, what we previously saw in previous works. Then, when we did a multidimensional analysis of the communities to understand the structure, what we saw is that, of course, there is a relation between samples coming from the same retreat area, even if they're distant one from the other. But if you have a look into the dendrogram, what we saw is that samples of older soils of all 3 retreat areas are more common among them, or more similar among them, than compared to the others from their own region. So we decided to have a look and ask and have the answer of how is this going on. And we saw that which abiotic factors and biotic interactions were having an impact in the structure of the cyanobacteria community. The first thing we saw is that pH was the only significant predictor in terms of abiotic factors of the community structure, explaining around 46% of the variation. And then the biotic interaction, it was acidobacteria, Armatomonaditis, and Verrucomycorrhea who were significant predictors, explaining 60% of the variation. This makes sense since we saw that the soils are not really developed, so probably the chemical features of the soils are not really defining the structure of these communities, but the biotic interactions. Anyway, it is necessary to do further studies to understand if maybe the eukaryotic portion not studied in this work could be or could have an impact also in the structure of the cyanobacterial community. And one thing that really surprised us, it was that we obtained a total of 315 ASPs, and 14 of them were present in every single sample we sequenced, not only those coming from the retreat areas, but also those coming from the cyanobacterial maps. And which is more, they have a relative abundance in these retreat areas between 52 and 82%, which is a really high relative abundance. Who were taking part of this core cyanobacterial community? Well, a total of 8 genera. And in the GenBank Better Hits said that it was phylotypes from Antarctica, the Arctic, and different biotypes from Europe and Asia. What is this telling us? That all these organisms first were exopolysaccharide producers and psychrophiles. So that those are cyanobacteria that can cope better with the Antarctic environmental conditions. Also, taking into account that this is stable because it's everywhere, and dominant taxa because it's high relative abundance, maybe the plasticity is the real key to withstand the different abiotic and biotic variables that control the composition of the study soils. Thus, our main conclusions were that our results confirm the role of cyanobacteria as key pioneer organisms in glacier retreat areas from Antarctica, acting as phototrophs and diazotrophs in these newly colonized soils. Also, that sequence assigned to filamentous non-heterocystic cyanobacteria dominated the 3 locations. As well that while chemical properties of soil remained almost stable with the range of age investigated, the interaction with other microbial communities appeared as a fundamental factor in determining the structure of cyanobacterial communities. And finally, that the presence of a core community with high relative abundances suggests the existence of a central community essential in processes of primary succession of recently deglaciated soils, and as well in the conformation of microbial nets. Thank you all for your attention.
Thank you, Maria Cristina. And now we move to next speaker, who is Laura Zucconi. And the title of the presentation is Effect of Endolytic Colonization on the Bacterial Diversity of Soils in Victoria Land. Please, Laura.
Okay. Do you see the presentation?
Yes, we do.
Okay, thank you. Thank you to the organizers, and thank you to everybody that is here listening to me. I'm going to tell you something about the bacterial diversity of soils in Victoria Land. Antarctica has different type of soils that are present. Soils without ice cover are present in continental Antarctica and on isolated mountain peaks and nunataks, or in the McMurdo dry valleys. And these soils have been assumed to be sterile until the 90s. Instead, we know today that these soils host a microbial community, a microbial community that is partly due to endolytic communities. The endolytic communities were described in 1987 by Imre Friedman, that you can see on the left side in the picture together with Silvano Onofri, was described as the last refuge for microorganisms when the environmental conditions are too extreme for life. And Antarctic relic soils are originated from rock weathering due to the growth of the endolitic communities, as well as to physicochemical alterations, physical chemical rock alterations. The growth of microorganisms cause an exfoliation phenomenon that is responsible of the release of small fragments that act as vegetative propagals, but in the same time falling into the soil contribute to the soil diversity. So it is reasonable to expect that the composition of soil microbial communities is influenced by species from nearby rock outcrops. Many studies have been carried out by metabarcoding approaches describing the communities of these soils, but we actually don't know exactly which is the contribution of DNA from living cells and which is the contribution from DNA of dead cells, of cells with damaged membranes. And moreover, the presence of relic DNA has been denounced by many authors as able to persist in soils in the dry and cold conditions of the Antarctica for very long time. So the matter of relic DNA and the estimation of rock and soil diversity has yet to be resolved. So which are the questions of this study? Are the microbiomes of rocks and the soils around them similar? Can rocks, which are thought to be microbial hotspots, contribute to soil microbial diversity? Which role does extracellular DNA play in the observed diversity of rocks and soils? And finally, do microorganisms survive in soils which lack that protected niche that characterizes rocks? To answer to these questions, we collected both soil and colonized rocks in 3 different localities in Victoria Land in triplicate, and the soil samples were collected at 3 increasing distance from rock outcrops. This is a picture of Battleship Promontory where there is a sandstone outcrop, and under the surface of the soil you can clearly see as the soil is composed from the alteration of the surrounding sandstone. For each sample we obtained 2 subsamples, and one of the 2 was treated with propidiomonazide. That is a compound that in presence of light binds the DNA of cells with compromised cell membranes and free DNA, inhibiting PCR amplification. It doesn't bind to living cells or to cells with intact membranes. DNA from both native or untreated samples and treated samples was extracted and used to characterize bacterial diversity through a DNA metabarcoding approach. These results were also analyzed, correlating them to the soil physicochemical parameters. Going to the results, the first interesting result is the higher diversity in soils than in rock samples. That was quite surprising. The second interesting result is the high contribution of rock in soil diversity, because a great number of the ASV present in rocks are also present in soils, where they represent, of course, a small portion of the total diversity, but what we have observed is that these are a small portion of the total soil diversity, but some are very, very abundant in soils. The same results were also obtained when the 3 localities were analyzed separately. These are results obtained with no treated samples. When we go to see the ASV distribution at increasing distance from the rocks in no treated samples, but we obtain more or less the same results also with treated samples, we observed a higher number of ASV shared among the 4 different types of samples. We observed the highest ASV shared among the soil samples, excluding the rocks. And we did not observe any correlation, any significant correlation with the distance. Going to see the ASV distribution in native and treated samples reported in red and in blue respectively, we found as a first interesting result a quite high number of ASV unique of to non-treated samples. So these are supposed to correspond to extracellular DNA or DNA of dead cells that is not amplified after the treatment. The highest number of ASV was obviously in common between treated and not treated samples. We would expect to have no ASV unique to treated samples. But even if these ASV were present, they were the lowest portion of the data set. And in general, ASV unique in treated samples with the PMA showed a very low abundance in the total data set, letting us to suppose that they are rare species whose presence may be offset by the presence of high amounts of extracellular DNA. When comparing the ASV distribution in treated rock and soil samples, we obtain more or less the same results that I already showed you for untreated samples. Looking at the richness in rock and soil sample, native rock soil sample, so untreated, we obtained a strong difference in the total richness between native rocks and soil samples. The same results were obtained when we analyzed treated rock and soil samples, but we here obtained just a little lower richness in soil, letting us think that in an open environment, as the soil is, the contribution of extracellular DNA may be higher than inside rocks that are more closed and protected environment. And finally, comparing the richness in PMA treated and treated samples, treated samples at increasing distances, we did not observe any significant differences correlated to the distance, but again, a difference in the richness values between rocks and higher richness in soils. Finally, when we try to investigate the possible influence of different physicochemical parameters on the richness of the main phyla, in both treated and treated samples, we obtained an interesting results. As you can see, in untreated samples, we observed only just a few positive and negative correlations, but what was surprising is the number of both positive and negative correlations that we obtained when the correlation was done using, analyzing samples treated with PMA. And this not only suggests the influence, the importance of environmental parameters in shaping the structure of these communities, but also suggests the risk of incorrect interpretation of soil analysis when no distinction is made between DNA from intact and damaged cells. Finally, looking at the composition of rock and soil communities, we can see this here. I'm showing you data from no treated samples, but from treated samples, the results were similar. We obtained a strong diversification stratification between rocks and soil communities, the first with triangles and the second with circles. Rocks of the three sites were well differentiated from each other, while no differences were observed among soils, neither when we observe the soils at different distances. So take our message as rocks contribute to soil microbial diversity, and soils have a greater diversity than rocks communities. Edaphic parameters affect the composition of soil communities, but of living soil communities. For rock, we have not yet the data. Extracellular DNA may be preserved in high proportion and can offset the estimations of microbial diversity and may offset the effects of environmental parameters. And finally, extracellular DNA plays a stronger contribution in soils than in more protected rock niches. Thank you for your attention.
Thank you so much, Laura, for your presentation. And next speaker is Matías Vargas Reyes, with the presentation use of weed-resistant bacteria isolated from Union Glacier, Antarctica for hydrogen photoproduction mediated by the biosynthesis of quantum dots and oxygen-tolerant Nife hydrogenases.
Hi, everyone. Can you hear me?
Yes, we can hear you.
Do you see the presentation?
Yes.
Yes.
Okay.
Perfect.
Well, my name is Matías Vargas Reyes. I'm a PhD student in bioinformatics and system biology from Andrés Bello University. And now I'm going to talk about to you on my research in Antarctic microbiology and biotechnology. Antarctica is the coldest and most arid continent on our planet. Environmental conditions vary widely across the continent, and even in certain areas, environmental conditions become more adverse. One hyper-extreme area of Antarctica is the Union Glacier. The Union Glacier is located 1,000 kilometers from the South Pole. Despite having low temperatures throughout the year, the presence of some bacteria has been reported, which is extremely interesting because of the environmental conditions of the Union Glacier. The Union Glacier has permanent radiation at surface level during the summer months due to the onset of night. In addition, radiation is increased at surface level by the effect of albedo and low cloud cover. Therefore, the permanent radiations reaching the Union Glacier will be causing changes in bacterial communities, in which bacteria are possibly developing molecular strategies to counteract the effect of the radiation. On the other hand, Union Glacier has a low organic matter content. As can be seen in the table, the percentage of carbon and nitrogen are lower than those reported in other hyper-extreme areas of Antarctica, such as Dry Valley. Researchers have proposed several ways in which microorganisms could be surviving nutrient scarcity in areas with low organic matter content in Antarctica. One of these ways is through the uptake of atmospheric gases, such as hydrogen. Genomic analysis of bacterial communities have revealed the high presence of hydrogenases, enzymes that perform the reversible catalysis of hydrogens from electrons and protons. Specifically, the analysis revealed the presence of a group of oxygen-tolerant hydrogenases called group 1H. Hydrogenases have a great biotechnological potential since they can be used for hydrogen production. The environmental conditions of the Union Glacier have led the bacteria to generate molecular strategies to resist radiation at surface level and to possess enzymes such as hydrogenase to survive a low content of organic matter present in this area. In this sense, The bacteria that inhabited the Union Glacier have great biotechnological potential. Specifically, biotechnological applications for hydrogen production have gained great relevance in recent times. Recently, the favoring of hydrogen production through nanohybrid system has been described. These systems consist in the transfer of electrons from the fluorescent semiconductor nanoparticles or quantum dots irradiated towards hydrogenous enzymes for hydrogen catalysis. Our group has conducted extensive research on the biosynthesis of quantum dots in Antarctic bacteria. Quantum dots are nanostructures with sizes between 2 to 20 nanometers. The biosynthesis of quantum dots in bacteria consists of addition of metal ions, like cadmium chloride, in conjunction with a sulfur source, usually cysteine, leading to the formation of intracellular quantum dots, as you see in the 10 images, and fluorescent pellets, and extracellular quantum dots. Quantum dots, when are irradiated, usually in the UV range, can transfer the electrons to other molecules. Therefore, due to the environmental conditions of Union glaciers, UV-resistant bacteria, which present hydrogenase and which can biosynthesize quantum dots, will be living there. Based on this, the Union glacier bacteria will be an excellent model to be used as a nanohybrid system for hydrogen production. Based on the background present, the objective of this work was to evaluate the potential use of UV-resistant bacteria isolated from Union glacier for hydrogen production needed by the biosynthesis of quantum dots and oxygen-tolerant hydrogenases. The samples were taken from the Elborz Mountains on Union glaciers, thanks to the Antarctic scientific expedition organized by Chilean Antarctic Institute. Soil samples were taken from Grossman Cove and Elephant Head. Bacteria were isolated from soil samples using R2A medium at different incubation temperatures and were identified by 16S sequences. The most predominant bacteria were the genera Paracoccus and Arthrobacter. UV-resistant bacteria were selected by exposing them to the lethal UVC doses of E. coli. Bacteria that showed a higher percentage of survival were subjected to higher doses to UVB and UVC radiation. As shown in the figure, bacteria of the genera Arthrobacter and Paracoccus showed resistance to different UVB and UVC doses. Bacteria of the genus Arthrobacter biosynthesize quantum dots in the presence of cadmium chloride and cysteine, specifically cadmium sulfur quantum dots. The formation of quantum dots can be evidenced by fluorescent emissions from the bacterial pellets. In addition, bacteria on the genus Arthrobacter biosynthesize extracellular quantum dots. In turn, most bacteria of the genus Arthrobacter and Paracoccus biosynthesize extracellular cadmium sulfide quantum dots at 24 and 48 hours using cadmium chloride concentrations of 0.5 millimolar and cysteine concentrations of 1 millimolar, where fluorescent pellets are seen in the figure. Genomic analysis of Paracoccus RC21 bacteria revealed the presence of putative nickel iron hydrogenase and NADP reducing hydrogenase. On the other hand, protein analysis of Arthrobacter EH1B1 revealed the presence of putative nitrogenases, enzymes that, like hydrogenases, can catalyze hydrogen evolution reactions. In conclusions, UV-resistant bacteria were isolated from Union glacier soil samples. These bacteria were able to biosynthesize intracellular and extracellular cadmium sulfide quantum dots. Finally, putative hydrogenase and nitrogenase were identified in the genomes of 2 UV-resistant bacteria. Currently, we are evaluating the photoproduction of hydrogen by irradiation of UV-resistant bacteria biosynthesizing quantum dots. Unfortunately, however, in our lab, we don't have equipment for rapid screening to determine hydrogen productions by these bacteria. For this reason, we are looking for collaborators to complement this research. Finally, I would like to thank to Andres Bello University, the Center for Bioinformatics and Integrative Biology, and the Laboratory of Bionanotechnology and Microbiology that made possible the scientific development of the research, and the Chilean Antarctic Institute in conjunction with the Chilean Armed Force, which allow us to reach and take samples for the Union Glacier. Thank you very much.
Thank you, Matthias, and I hope with this session you will be able to find the collaborators for your project. Now we have time for questions, so if you have any questions, you can write it down in the chat box and we can make your question to the speakers. In the meantime, if there are no questions, I have one for Maria Cristina. It's just show that in the younger and older soils, there are the same orders that are dominant, while they decrease. The most dominant order decrease in the intermediate samples. Do you have an explanation for that? Because it seems that older soils are more similar to younger ones than to intermediate ones.
Yeah, that was something that was surprising for us, because what we would expect is something like a trend, right? Like that some orders that are at the beginning are less at the end, or the opposite. But what we thought that maybe it's happening as we saw also in the proportion of cyanobacteria related to the whole microbial community in these 2 retreat areas in Clark, Nunatak, and Elephant Point, is that maybe these soils are having these extra nutrients coming from somewhere else, because these 2 orders, or the role of the oscillatories, is expected to be like more important for carbon and nitrogen fixation. So it's something that we think that these intermediate soils, somehow we are not really controlling what's going on there, because otherwise we would expect that the more developed soils, or that the younger soils from both were closer even between them in the structure and so on, but we didn't see that. It's something that we have to go deeper into it.
Okay, thank you so much.
Sorry.
And we have questions from Byron Adams to Laura Zucconi. The question is, wow, the difference in the number of positive and negative correlation between taxonomic groups when the sample is treated with PMA is really large. Many of these are clearly due to soil properties, but given that only viable cells are being included in this analysis, could biotic interaction also explain some of these large differences? Laura, your microphone is off.
Sorry, I didn't understand the last part of the questions. So, what did you tell me?
You can read it, but given that only viable cells are being included in the analysis, could biotic interaction also explain some of these large differences?
Yes, I think that the reason of these differences is exactly due to the contribution of that part of the DNA that comes exactly from cells that are living cells or cells that are not damages. So what is interesting of the result is exactly this result, that the contribution of the parameters, the number of correlation is exactly determined by the living component of the soil. I don't know, is the answer, it's what he wanted to know.
Maybe if you want to ask us something more, Byron, we can open your microphone also if you want. Or if there are other questions for the speakers.
I have a question, but it's a very naive, I would say, because it's out of my expertise, and it could be for each of you because it's a very general one. Actually, it's about two questions, I would say. How do you, in your techniques, the methods you use to measure your parameters, how can you put aside possible contamination by pollution? And I would extend that question to the plastics and microplastics. Do you know if there is some clear interaction with these materials that could interfere in your results? It's too general, sorry.
I know that there are many studies concerning plastics also on soils, but I don't know how to evaluate the role of plastic and microplastics really on the microbial diversity. Pollution is surely a problem, and many studies have been carried out on the ability of some microorganisms to degrade different type of pollutants. And a colleague of us is working in this, actually working on the degradation of oils working with soils, for example, collected in Key Island, and she has obtained an incredible ability to degrade these compounds in a very, very short time. So there are some microorganisms that probably have are of great interest in the biotechnological application for different in different fields. But for plastic, I don't know.
Okay, thank you. There is another question by Adams for Maria. Did you notice that cyanobacteria diversity and or abundance decreases over time with ecological succession?
Sorry. Yeah, actually, about the abundances, we saw that it decreases, and that's what we explain as that their role as pioneer organisms makes no sense maybe in later periods of the succession. What is clearly visible in Biscuit Point and in the other two retreat areas, we see that even the abundance decrease between the intermediate and the older soils. And regarding the diversity that you said, sorry, I have here some data that I didn't include. It's not always less diverse. Sometimes we saw there is a shift actually in the between different soil ages, but we didn't see a trend that older soils have less less diversity. We didn't see that trend in any of the soils.
Okay.
May I add just I think, if there is time enough.
I'm sorry,
no.
Laura, we have to move to next speaker. It's time to next speaker, but maybe later at the end, you will have some time.
I just wanted to say that I was thinking to the first question that we are also waiting for the results for the fungal component of these communities. So it would be interesting to see if the same correlation and positive and negative correlation with the soil physicochemical parameters will be confirmed for the living component, fungal component of the communities. Just so. Okay, thank you.
Thank you, Laura. And next speaker is now Sabrina Heiser. And the presentation is what drives the distribution of chemical defense diversity in a macroalga?
All right, can you see my screen?
Yes, we do.
Thank you. All right, so thank you very much for this opportunity to present. I'm very excited to present part of my PhD research here. Sessile organisms in general are at a slight disadvantage when it comes to things like evading predation, biofollowers, or competitors, since they aren't able to simply move away from the danger. So that means that they either often have structural or chemical defenses instead, or some of them might even have both. And when it comes to the chemical defenses, those are generally the secondary metabolites because they're not involved in the primary metabolism of the organism. And for the defenses, whether they are structural or chemical, we can often observe diversity between or even within species. And that in turn can impact the success and distribution of the producer as well as associated organisms. And I'm interested in understanding the distribution of those phenotypes in order to better understand ecosystem structure in general. And we know that phenotypes in general may be determined by the underlying genotype of the organism, by the environment, or most likely an interaction between the two. The genus of my study organism, Plochemium, is known to have a worldwide distribution, and from that we know that it's high in secondary metabolite chemodiversity even within populations. And it has been extensively studied here at Palmer Station on the Western Antarctic Peninsula. And we knew when we started this study from a small previous study that there are at least 5 different chemogroups within the area. So what's a chemogroup? A chemogroup is basically a signature mixture or a signature cocktail of different secondary metabolites. And you're going to see me color code these throughout the rest of the talk to visualize the diversity. And those chemogroups differ in the particular compounds that are present. So I've got just 4 random examples here. So they differ in the particular compounds that they have, but they might also be producing the same compounds, but as you see with the height of the peak in different relative quantities. And in our field seasons between 2016 and 2018, we chemotyped over 1,400 different thalli, and we identified 15 different chemogroups from the area, which we arbitrarily named A through O. The other observation that had been made in the initial study that I introduced earlier was that certain chemogroups were found at certain sites, which indicates that there is an underlying geographic distribution of those chemogroups. And we were interested in whether there are any fine scale within site patterns that hadn't been picked up on before. And in order to look at that, we conducted transects perpendicular to the shore. And I'm now going to show you the results for 5 of the sites where we were able to run 3 full transects for each of the sites. On the bottom here, we have the chemogroups that we found at those particular sites because we didn't find all of the chemogroups at all sites. And as I mentioned, we ran 3 transects at each of the sites, but when we compared the transects among each other, we didn't find any statistical significant difference between those transects. So what I did, that's why I combined all 3 transects on one plot, which is easier to look at the data. And what we have here is on the vertical axis, we have the depth distribution. We sampled at 3-meter depth intervals. And then on the horizontal axis, we have the number of thalli that we collected. And we tried to always collect 15 thalli from each of the depths, but that wasn't necessarily possible when Plochemium wasn't necessarily always abundant enough. And when we just look at all of the sites, we can see they're dominated by different chemogroups. So that kind of confirms what the original study found, and those differences were statistically significant. And we also saw some differences among the depths for some of the sites. So we can see at Nozzle Point here, for example, the shallower depths are dominated by different chemogroups than the deeper depths. So now I will talk about what this chemogroup diversity means to the organisms, and then I will end on what is potentially driving the observed distribution that we have here. From previous research, we know that the Antarctic Plochemium is very heavily defended due to those secondary metabolites. And there's in fact only one known grazer that can feed on it. And that's this little amphipod here, Paradexamine piscicauda, which is also more frequently found on Plochemium. And it's the only amphipod that we know that has this skewed distribution. But we also know that it does feed on other algae as well. So it's not a specialist per se. So the question is whether chemogroup production impacts the only known grazer or its producer. In order to look at that, we conducted feeding rate experiments where we had single chemogroup treatments, which you see on the horizontal axis here. And we picked the chemogroups that were basically abundant enough in nature so that we would have enough replicates. And then on the vertical axis, we have the feeding rate of the amphipods. We also use Palmaria decipiens, which is not chemically defended, and we know that Paradexamine will readily feed on it as a comparison. And you see by the different letters on top of the box plots that we do have differences among the chemogroups, and some of them were clearly consumed at a slower rate than others. So it does matter to the producer as to what it's making for its defense. But what about the grazer? So in order to look at the impacts on the grazer, we conducted growth rate experiments where we kept the amphipods on the same feeding treatments that I just showed you in these big flow-through tanks on single treatment diets for about 60 to 65 days in these little amphipod hotels that we made. And when we looked at the growth rates, we didn't really see any clear patterns. We were also able to look at the reproductive output for a subset of the treatments. It was actually quite difficult to find amphipods that were big enough where we were sure that they're sexually mature. So we weren't able to run as many treatments and as many replicates. And when we compared the number of gravid females, again, we didn't see any differences among the treatments. The amphipods, Paradexamine, they brood their young. So you see the underside of the amphipods here, which also allowed us to enumerate the number of embryos that they were producing depending on the feeding treatment. And this is where we did see a significant difference. So we have chemogroup L here, which had significantly fewer amphipods than the ones that were fed on the non-chemically defended Palmaria. Chemogroup M, as you see, was not significantly different from either of the treatments, but it did have fairly large variation. And due to the constraints that I mentioned earlier, we had a fairly small sample size. So overall, some chemogroups were consumed at a slower rate. There was a strong effect on the reproductive output, and we saw variation between chemogroups and individuals upon which selection might be acting. This also raises the question whether differences in grazing pressure and maybe grazer abundances could have an impact on the distribution of Plochemium and its chemogroups. So as I mentioned earlier, we see this strong geographic distribution of the chemogroups, and the question is whether the environment, whether biotic or abiotic factors, underlying genetics, or an interaction between the two is driving the distribution of those chemogroups. So to test whether the environment is driving this, we picked 3 of the sites that I showed you earlier to conduct reciprocal transplant experiments to see whether Plochemium could change within its lifetime what it's producing. We transplanted them in 2017, came back in 2018, and happy days when they weren't wiped out. None of them were wiped out by any icebergs. But not only that, you can also see that Plocaemium had grown, so it was doing well. When we analyzed what chemogroups they were producing the year later, we didn't see many changes at all in the chemogroup production. So that overall indicates that the environment is not driving it and the distribution of the chemogroups, at least within the lifetime of the thallus, which indicates that there are underlying genetic differences that are driving the distribution of the chemogroups. We did single gene sequencing where we found two main haplotypes. Those are the same that had been identified by the small original study that I introduced in the beginning. And what we have here is a haplotype network, and you see that we also found a couple of additional haplotypes. The different colors indicate the different chemogroups, and we see that they sold themselves quite well into those two main haplotypes. However, we needed more polymorphic markers in order to look at the genetic differentiation and identify individuals in order to see whether there's any correlation between that and the chemogroup distribution. So we developed 10 microsatellite markers in order to analyze the same samples that we collected along the transects. And the first thing that we need to do is, was to describe the reproductive system of Plocaemium, which I don't have time to present today. And we needed to do that first because Plocaemium is a red seaweed and it has a haploid-diploid life cycle, and our haploid and diploid stages look identical. So unless they're reproductive, we're not able to tell them apart. And that will then allow us to describe the population structure. What we were able to do now is to look at our vegetative samples and see whether there are haploids or diploids. So the diploids are the reds, the tetrasporophytes, and then the haploids are the blue, the gametophytes, and the lighter shades are the samples that were vegetative when we collected them. And what you see here on the horizontal axis, again, we have the chemogroups and then the proportion of that was produced by tetrasporophytes and gametophytes. And even though we have lower numbers of gametophytes, we do see that no chemogroup was exclusively produced by gametophytes. And these here, I and J, were only produced by tetrasporophytes, but they also had very low sample sizes. So overall, life cycle stage is likely not correlated with the chemogroup production. So in summary, Plocaemium has a high chemodiversity that varies with geography and with depth. We see that chemogroup production matters to the seaweed and the grazer. It is not driven by the environment, at least within the lifetime of the thallus. And we see a potential correlation between the genetic structure and the chemogroup production. So what can we take away from this? This helps us understand why Plocaemium is as widespread and as abundant as it is within the area, as well as along the Western Antarctic Peninsula. It will also help us to understand better about the connectivity among Plocaemium populations, since there are no flagellated life cycle stages or spores or gametes. This is a really interesting question. And it will help us understand more about the drivers of the phenotypic distribution in sessile organisms. And with that, I'd like to acknowledge everyone, and I'm looking forward to questions later on.
Thank you, Sabrina, for your presentation. And next speaker is Santiago Zarate with the presentation Novel Pigments and Biopeptides in Marine Fungi from Antarctic.
Thank you very much. Can you hear me?
Yes, we
Okay.
can.
Yes.
Okay, I'm going to share my screen.
Okay.
Okay, let me know if you can see my presentation.
Yes, we can.
Okay, I'm going to put in the...
You just have to put to present, yes.
Okay, first of all, greetings from Ecuador. I'm going to try to speak these few minutes about the research involved in fungi from Antarctic, right? It's a big group that we're working on in different areas, basically peptides and also for pigments from fungi. Let me introduce briefly our team. a lot of contribution, especially with two research from Ecuador, using all the data from the Pedro Vicente Maldonado research station. Well, why Antarctica? The whole of the day you are see, and also listening the idea the why we're using the information, also the biodiversity of the Antarctica, but as you know, all the factors that we have in this beautiful continent are the more suitable for different conditions, right? In terms of the UV radiation, photoperiod, strong wind, desiccation, extreme temperature, and low nutrient availability. Therefore, all these factors trigger all the metabolism in different organisms, in this case, the fungi, for producing the types of polymers, peptides, pigments, and enzymes. In the case of a group, we're really focusing on peptides and pigment. I'm going to see why it's important to do that. Okay. As you know, during the days, the conditions in the continent, fungi always are fighting with other microorganisms in terms of bacteria, right? So pigments in terms of the fungi are really focusing on terms of protect the UV radiation and also for bacterial attack. Therefore, we can find different pigments from fungi and different colors. They are very different from the plants because plants has chlorophyll. But in this case, we're really interested in terms of anthocyanin, beta-carotene, riboflavin, astaxanthin, monacin. Then we can change all the chemicals that really involve in terms of pigments for food and also for textiles, in terms to reintroduce these biological pigments for the same sectors as a food, cosmetic, and textiles. And the other way, we can also take about the peptides. Peptides, as you know, is related to short molecules from small chains of amino acids between 18 and 24 amino acids per chain. But the interesting thing is that peptides exert different activities. For example, antioxidant, immunomodulatory, anti-cancer, antimicrobial, anti-inflammatory, liver disease treatment, lipid lowering, and so on. So therefore, if we can focus it on in these two main areas, we really want to go to industrial biotechnology to produce this in large scale. So basically, peptides and pigments are really important in terms of the fungi because in the surrounding, they want to fight with all the conditions, not only for the biotic stress, but also with an abiotic stress. So therefore, we believe that these peptides and these pigments could be a good solution, try to move from the chemical dependence to biological dependence. Okay, all these fungi where I'm going to talk in this short meeting is related, came from the three basic areas. As you can see in the slide, is then the surrounding of the Pedro Vicente Maldonado in the Culebra River, right? We also have some collections for the Isla D Island and also Barretos Island. Okay, the sampling was carried on by two of colleagues. See, this sampling was taken in terms of all the conditions to transport the samples, avoiding some contaminations. But as you know, you have to transport all the samples to the lab. So we have to transport the samples to the Ecuador. So the way is so far from the south of the continent. Therefore, after we can take the samples, we will also name it, but the code, and also wait after the transportation, we initiate the research. In terms of the methodology, you can see we start with a isolated strain, which is a big part because most of the samples were taken over 4 and 8 degrees Celsius. So in the lab, we try to adapt to the temperature basically, and also to the conditions. The isolated strains went to a submerged fermentation in different medias in terms of pH, carbon source, and temperatures. There, we proceed to extract and concentrate pigments and peptides in different ways in terms to then analyze the bioactivity. First of all, we characterize in terms of molecular characterization by ETS sequence. Then for the molecules, basically for proteins, we characterize in terms of weight size profile through DSA space and magnitude. For the pigments, we use chromatography, and also for pigments as well, UV spectroscopy. Moving on, after to see what's going on with the molecules, we see what are doing in terms of bioactivity and toxicity. Here I want to show you a small results because we are waiting for the publication, but basically we focuses on terms of anti-nematode activity with cell elegance survival, and also with pigments antioxidant activity by DPPH method for terms of the pigments. And for biopeptides, we focused on antibacterial activity, mainly for resistant bacteria in terms of Staphylococcus aureus, E. coli, and Klebsiella pneumoniae. Well, what about the briefly results? In terms of the pigments, we can see that one strain, named by UTN AA17, most of them exert a nice effect over C. elegans. As you know, in the orange mark, you see how these strains develop almost 30% of survival compared with another strain and also with the controls. In the case of Ecuador, this is interesting because we are really dependent of the agriculture. So we are really focusing to see which methods could be help to us to control the pests and disease on the plants. So maybe this pigment could be a good approach to see that. And the other way around, we also evaluate the antioxidant activity with the DPPH4 method. The same strain exerts like a percentage of over 85% of antioxidant activity. It was expected because most of the fungi in the Antarctic exert these pigments to control or to tolerate the UV radiation. So you can see move using this pigment in terms of cosmetic or nutraceutical applications in terms of the protection of UV, right? What happens with the small peptides? Well, after to concentrate the peptides and also to apply a small dialysis between them, we see that two strains exert a nice inhibition halo in terms of the development of the bacteria. In this case, for example, this strain, utinane AD31, yeah, the peptides that flank the range between 10 and 100 kilodaltons of molecular weight exert this kind of inhibition halo in in Staphylococcus aureus, right? Also, we try to see if the small peptides over less 10 kilodaltons of weight say exert the same effect, but you can see that there is not effect at all. And also we can see the same effect with another strain in terms of the radio of the halo. Now, it's interesting this brief result because we can use these peptides in terms to control and also to develop new techniques in terms of antibiotic field. Basically, in Ecuador, we have another problem. Well, I think all the world have the problem in terms of bacterial resistance. So we also really focusing on to know new molecules that could be the solution in terms to take over this problem. So until now, we are focusing more in butane A10 because, as you see before, not only for pigment but also for biopeptides, it has a nice results, and also we can go in another level, right? Okay, finally, in terms of future perspectives, we really want to scale the process. All the experiments were made in lab scale, but now we're going to go to pilot scale and also for industrial scale, based on terms of pigment and peptides. Here in the presentation, I only introduce you the best results in terms of two strains, but we have another strains, right? One of the big questions always when we are producing this kind of molecules is if the temperature has a particular effect in terms of exert this activity. Yeah, totally right, because the temperature in the south is coldest. But if we want to go to industrial scale, we have to adaptate these organisms to other kind of temperature in terms of decrease the energy demand of the production. Finally, I would like to say thanks to all the people that were really involved in, first of all, in terms of the university, all the research groups, and also for the Army of Ecuador, INAE, and also the expedition where our colleagues were taken from. So thank you for your attention. If you have questions, just go ahead. Thank you so much.
Thank you so much for your presentation. The next speaker is Paul Sekowski. I hope the pronunciation is right.
That's all right, thank you.
Thank you. The presentation is Antarctic biodiversity predictions through substrate qualities and environmental DNA.
Okay, now share screen. This one. Share. And okay, you should see my screen.
Yes.
Thank you very much for having me. It's my first virtual conference, interesting. So today I want to talk about something that I've been doing for the last 10 years, and it's one of my most favorite projects. And I've analyzed this data a couple of times, but the final goal really I achieved just now, and the paper is just coming out. So everything I'm talking about, you can read. It's coming out in Frontiers of Ecology and the Environment, ESA. Let's see if I can change slides. I'm here in Leipzig right now. It's my hometown. My last project was ancient DNA. I'm also working in ancient DNA and environmental DNA here in Argentina. But of course, my big passion is Antarctica. So I work a lot. I like working a lot with Antarctic invertebrates as the tardigrade here. And doing my PhD, which was 2012 to 2016, I had this amazing opportunity to adopt environmental DNA service to Antarctica. And basically, I wrote a review about it, where this figure is from. It entails going to Antarctica, taking soil, getting the DNA out of the soil, and then looking by the sequencing what kinds of invertebrates are in there and relating them to the environment. Okay, so that's not modern technology anymore, but I'm just repeating it anyways. So I went to East Antarctica. The grant was attracted by Mark Stevens of the South Australian Museum, and was supported by the Australian Antarctic Division. We visited 3 locations in the Prince Charles Mountains for 60 days. It was very remote locations, and we investigated these ice-free areas there. And took soil samples everywhere. So one of the main goals of this project was to be able to predict where we can find invertebrates, so that one may target where to look for them and identify species that are worthy of conservation, or areas that should be on which conservation efforts should be focused. And I tried doing my PhD using various older techniques, and only now it has been possible for me and my team to do a newer analysis technology. And here we see the whole sampling at Mount Menzies. I'm going to talk about the red spots later, but everywhere where you see a circle. So here's Antarctica, here's the Prince Charles Mountains. There are 4 maps where we collected the samples, so just that you can see where we went. Then I used an 18S marker to generate environmental DNA libraries, and there's sound now. Watch your ears. So it's a robot. If it plays, does it play? Yeah, it plays. So I used robotic pipetting, which was fun, and to do that, and only this way I could pipette all these plates cleanly. And I would still advertise doing that if we have access to a robot. Then I used Chime, and I learned it, but I now, you know, everything that Chime doesn't implement, I can do using Bash or my own R scripts to do anything that Chime doesn't provide. So we use the full NCBI data with a negative GI list to exclude all environmental sequences, to annotate those taxonomically. Let's see. And then looked at all the taxonomic assignments in Megan. And here's an example for an Antarctic nematode and one of the query sequences from the Prince Charles Mountains. And there's one gap. So we can't be sure that it's actually that Antarctic nematode. So just to be sure, we only looked at the phylum level. So we only dealt with nematodes and tardigrades and such higher taxonomy units. And then the main takeaway of this talk is that we found out how to relate the main invertebrates to the environment where they are. So here you have a map of how many different OTUs we found at each location, and on the right side, what predictors are most relevant to the presence of these taxa. Among the predictors were standard soil geochemical variables that can be collected by any agricultural testing lab, and X-ray spectra of the bedrock. So, for example, for the nematode, this was conductivity, and the presence at Mount Menzies, just the dry location. For the tardigrades, it was also conductivity and pH value. And for some fungi, it was also pH value. Again, only the location for chlorophytes, or again, the pH value for celiophores, garnets, and maybe sulfur. And so by using that technique, lasso regression, we were able to put many predictors and relate many predictors to the occurrence data of all these invertebrate groups. And then by repeating each lasso regression 1,000 times, we were able to isolate variables that are contained in the active set over 1,000 times, and therefore we were able to determine confidence intervals and these predictors which are relevant. And just as an overview, now I come to the red spots. Another aspect of this work was also to show that even in the coldest and windiest places of the world, I've never been to a colder place than Mount Menzies down here in this valley, there can be a surprising hotspot of biodiversity. So what I basically plotted down here as the red spots is just how many life forms, the diversity, how many we found at each of these sites. So we can easily map out highly diverse spots. So where the text summaries will also be in the manuscript. The text is down here, and of course, the text is in the talk. If you Google my name, you'll find this coming out soon. It's somewhat related to that earlier paper that is in the footer here. But basically now, if we have a data set of mineral composition and soil predictors, we can now completely map out in the Antarctica and tell at which locations we likely will find nematodes, tardigrades, fungi, and so forth. And of course, all this work is not only my work, but these 5 people who've been with me. So they did a lot. Fiona, Adrian, Nick, and Josh here. And we also, going north, we had lots more visited from wildlife. And of course, it all wouldn't have been possible without the AAD and without the support of Mark Stevens, who had the idea and got the grant, whom I don't have a picture of. So yeah, that's me there. Thank you very much. If you have questions, let me know. Thank you very much.
Thank you so much, Paul, for your presentation. Actually, we still have a couple of minutes before next presentation. So if you want to add something, please feel free.
to 36. 2 more minutes. No, I have lost the slides. Yeah, if anyone wants to ask me some questions, I can answer them now. I just try to make it as brief as possible.
Actually, we will have all the questions at the end in the question and answer session.
Yeah, no, I understand. Okay, so okay. Maybe we can. Oh, we can't move on. Well, if I can talk further. Well, so there's I'm in the paper. There's going to be a thorough presentation of which factors are associated with a probability of which taxa are at each of the which taxa are associated with what predictors. And so what you get is a probability map. So if you have a certain set of predictors and you see the measurements of soil geochemical variables and mineral composition, you can say, well, there's an 88% chance that we find a fungus at this location. So that could be useful in species distribution modeling of all of Antarctica, or other such modeling approaches, or creating maps, of course, then and then zoning areas of Antarctica according to their likely abundant biodiversity, as Alex Tiros does from the Australian Antarctic Division.
Okay, thank you. And now we can move to next speaker. The last for this session is Amy Moran. The presentation is Who is Watching the Kids? An answer to the century-old mystery of colossal sea spider production.
All right, thank you. Can you hear me okay?
Yes, we can hear you.
Okay, great, thanks. So what I'm going to talk to you about today is some recent observations and some sort of new discoveries that we made in just this past summer at McMurdo Station, Antarctica, answering one of the sort of long-standing questions in sea spider reproduction. So sorry, I just put something in the chat. There's a video that's going to come up later on, and I realize you may not be able to see the video when I play it through Zoom. So please click on that link if you want to see it. So for some background, sea spiders or pycnogonids are an enigmatic and ancient class of arthropods. They have a worldwide marine distribution, and there's about 13 to 1,400 living species, of which about 20% are Antarctic. So they are an important group in the Antarctic and diverse and comparatively abundant. The family Colossendiidae is the group which contains perhaps the most charismatic of all sea spiders, has the largest and the most conspicuous species in it, with many examples of both polar gigantism and deep sea gigantism. This is the group that has the classic dinner plate sized sea spiders. There's about 40 species in this family in the Antarctic, and there's a long history of research and collection of these species in the Southern Ocean, going back to the early 19th century. So the Colossendiidae are very common. They're often observed. So this is a photograph taken right off of McMurdo Station, Antarctica, at about 90 feet. And you can see they're abundant in this shot and probably ecologically relatively significant. But one long-standing mystery in sea spider biology is that reproduction in this family is completely unknown. That's surprising, particularly in the sea spiders, because the sea spiders have a interesting and well-known type of reproduction. They're well known for having exclusive paternal care of offspring. So the father broods the offspring, and this is the rarest type of parental care in the animal kingdom. It works like this. So there's a lot of weird things about sea spiders, but one of the weirder ones is that their gonads are in their legs. And so if we zoom in on the femur of this female sea spider, you can see the eggs, which are these little orange dots. You can see them through the cuticle. When these animals mate, the males sort of stand on top of the females, which is why there are so many legs in this picture. The females extrude eggs through pores in their legs. The males release sperm, and then the males gather up the fertilized eggs and sort of package them with glue and then attach them to ovigerous legs, a specialized pair of appendages, and carry them around for weeks to years. So this particular species is an Antarctic species that we've timed their reproduction at more than 2 years. So these males carry their eggs for a very long time. And that means that when people collect these, when they show up in museum collections, the males very often have brooded embryos on them. That means that we know quite a bit about the diversity of types of brooding, male brooding in the sea spiders. The most common stage at which larvae are released from the male is this type 1 or typical protonymphon larva, which has a little pair of chelicerae, a pair of two pairs of larval appendages and a little proboscis. These are most likely free-living predators or parasites and benthic. And as you can see from these phylogenies, the type 1 larva is shown here in green, and kind of whichever way you construct the tree of sea spiders, that comes up as the most common and probably the ancestral phase. With other types of development having yolkier larvae or sort of more derived forms. So the mystery of this talk that I'm talking about is that no brooding animals have ever been seen in the Colossendae, and their embryos and larvae are completely unknown. And the first report of this that I'm aware of is from Hooke in 1881, from the report on the Pycnogonida dredged by the Challenger expedition. And he was talking about the Colossendae, noted that none of the collected specimens from the expedition had embryos on them, and said that it is possible that this genus is an exception to the sea spider rule, and that the males may not have the gallantry to nurse their babies as the males of the species of other genera are accustomed to do. So this point has been made many times in the literature since then, although not with that kind of poetic language. Just to pick out a couple of sort of major examples in the biology of the Pycnogonida, which is a large work by Arnaud and Bamber in 1987, 100 years after Hooke. They point out again that despite the large number of collections of these animals, no males have ever been found carrying eggs, and something else must be going on. Breneus et al. Repeated in 2017 basically the same point, but also pointed out that this is really astounding in the case of the Colossendae, due to how prominent they are in collections. So what I'm talking about today are some observations that we made as part of a much larger project, sort of an offshoot of a bigger project that we just finished up our field season, and the summer field season just in February of this year. And the project is looking at thermal sensitivity of invertebrates, and Graham Lobert and Aaron Toe, the two co-authors on this talk, actually gave talks on the larger part of this work just yesterday in the same session. So please check those recorded talks out. But all three of us had a sort of long-standing interest in trying to figure out how the Colossendae reproduced. So we were diving already, and we were collecting sea spiders and other invertebrates. And one thing that we had noted and that others had noted for a long time is that Colossendae are actually often seen mating in the field, or apparently mating. So fairly often you see them in this mating posture with one individual standing on top of another individual. So what we did, this was Aaron's idea, we would collect these mating pairs or sets, sometimes more than two, of Colossendae, in this case Colossendae megalonyx, which is the species that we were able to collect. And we would bring them back to the laboratory as sort of undisturbed as possible. And this is an example of a mating pair with a male on top of a female. And this is two males standing on top of a female. And we just watched them, basically. We just looked at them every day to see if we could figure out what they were doing and how they were reproducing. So we taught two different spawning events. We got different information from both of them, and I'm going to tell you about both. So the first one was on October 25th, and one of the mating stacks had disaggregated. And what we saw was a spider standing in the sea table amidst this big cloud of sort of gelatinous eggs. And the cloud was very sticky. So we grabbed all the eggs and we started monitoring them on the microscope. And we were able to do this from October of 2021 to now, thanks to support from NSF. And what we found were, we were able to describe the early development. The eggs are small, about 100 plus microns in diameter. They undergo holoblastic cleavage, 2 to 4 cell by 8 days. They progress through gastrulation, all very typical of other sea spiders. And then over the course of the next several months, we progressively saw development of larval morphology. until just actually like 2 weeks ago, they hatched. And so we can confirm that Calissendae eggs hatch after 9 months, or megalonyx at minus 1.8 centigrade, and that they have these type 1 protonymphon larvae, which are typical of the sort of possible likely ancestral state in the sea spiders generally, with a nice little pair of chelicerae and a proboscis and larval appendages. So that answered one part of the mystery, but where do these eggs develop? Since they're not brooded on the males, where are they when they're developing? And is there any sort of sign of post-spawning parental care that we could see? So these answers came from a different set of sea spiders. Again, this is one with more than one male standing on top of a female, which is fairly common. The same situation here. We were monitoring this group of mating individuals. One day we came down and the stack had disaggregated, and there was a spider standing close to a set of eggs that were stuck down to the rock that the group had been standing on. We left these in place, relatively undisturbed. We left the adults as undisturbed as possible, and we watched them to see what they would do if they responded in any way to that egg mass. And we made this really interesting observation, and this is the video that's in the chat. I don't know how well it's going to translate when I play it now. But this is one of those parents from the stack, we think a male. And what it did was stand over that egg mass for 2 days and kind of walk around on it. And it was manipulating the egg mass with its oviduras and its palps and its proboscis. And what it really looked like it was doing was sort of aggregating that egg mass and consolidating it into a flat sheet. Indeed, after a couple of days, this egg mass was tightly consolidated and really well stuck onto the rock. So 3.5 months later, this was the end of our field season, so we had to stop these observations. But 3.5 months later, this is that same egg mass. The eggs are still attached, benthic, and they're very cryptic. As you can see, these little spots here, those are the embryos. In this case, actually developing inside the empty houses of a dead bryozoan colony. So very cryptic egg masses. So just to summarize our findings here, Calissendae megalonyx has benthic development that lasts around 9 months. They hatch out as type 1 protonymphon larvae, which is the kind of most common type of development in the sea spiders. And we think that they probably lack a pelagic larval stage. We just don't see a sign of a pelagic larval stage in their life histories. This is the first observation of non-brooded development in the pycnogonids generally, as well as the first observation in the Calissendae. And we think that no one's seen this before because it happens pretty fast. And the egg masses are highly cryptic once they've been produced. Interestingly, once we knew what they looked like, we were actually able to find a couple of them in the field with an individual tending them. And if you're interested in knowing how to do that, please let us know. Lastly, possibly we see the male tending masses after spawning. So one of our next interests is thinking about this in the context of the evolution of paternal brooding in sea spiders generally. In this phylogeny of all the extant families, paternal brooding is shown in blue. The gray families show ones where paternal brooding was not previously known, and we've added the Calissendae as having non-brooded development to this phylogeny. These other two families, interestingly, have also been pointed out as never having brooding males. And so we would like to suggest that those, in fact, have non-brooding development. Paternal brooding may be derived from within the pycnogonids and may have evolved more than once, potentially. So I'd like to thank you all for listening. I thank the U.S. National Science Foundation for funding, the Antarctic Support Contract staff for all their help, the UH Safety Program, the SCAR organizers, and Fernanda, Eugenia, and Fabiana for organizing this session. So thank you very much.
Thank you so much for your presentation. And now it's time for questions. So if someone wants to ask something. In the meantime, I have a question, actually a couple of questions for Santiago. So you did that, you performed the molecular identification of fungi, but probably I missed it, but you have some information on which species they are those that resulted more promising in production of peptides and pigments. And the second one is that in my experience with Antarctic fungi, often we see that they are very slow growing. So you said that you were moving to bioreactor and industrial scales, but usually the slow growth of these fungi impair their use in industrial applications. So if you have some information about the speed of the growth of these fungi.
Thank you, Fabiana. Yes, I had to short the presentation, but in order to answer your first questions about the molecular, the main strains belong to Fusarium strains and also Trichoderma. So we are also waiting for more molecular results about the identification, right? But also we're trying to put just the name of the code. But in terms of the growth, yeah, totally right. At the beginning, we have very serious problems to force the speed, the growth in the lab, but we try to manage the carbon source. We put some different gradients of carbon in terms of malaise, and also for no glucose in analytical grade, right? Because if you want to go to industrial scale, you have to decrease the cost. But we saw that in terms of the malaise, also helped us to double the time in terms of the speed. We don't have results about the industrial pilot scale in terms of the speed of the growth of the fungi, but it's the next step. But really focused in terms of that. And also we see that we change the temperature because we start with a thermocam. We growth to 5 degrees Celsius, but also we have a slow growth, but we change to 22 or 23, and also with environmental temperature, we also triplicate the speed of the fungi. However, the pigment of the expression decrease, so we have to put more stress in terms of the media, also by pH. But at least it's all results now.
Thank you so much. And there is a question for Amy from Byron Adams. I may have missed it, but is there sexual dimorphism? In the photos, it looked like that males were typically smaller than females. Is the mate choice system known?
That's a great question. In the Calisandeidae, the family that we were interested in, there's very little sexual dimorphism. So that's why we're actually uncertain whether the ones are males or females yet. We still have to dissect them out and look at their internal anatomy. But in the other species, many of the other sea spiders where the males do brood, there is a lot of sexual dimorphism that's related to that brooding. So it's another just sort of circumstantial piece of evidence about how they reproduce. It's a good question.
Okay, so if there are other questions, I have a question for Sabrina. You showed us the mechanism of resistance of these algae, but do you know if they can have any industrial application, for example, in production of pesticides or something similar as we have seen, for example, for fungi?
That's a good question. I'm not 100% sure about pesticides and things like that. I know that some of the secondary metabolites, I think, have been screened for drug discovery purposes. And the problem is that they're too potent, so they kill the good cells as well as the bad ones. But I don't know for pesticides.
Okay, thank you. Any other question?
I have a question for Amy. Amy, is there a substrate preference for the individuals to have that behavior of aggregating the eggs, or they just use any kind of substrate, or they use some sort of rock or inconsolidated substrate? Is there any preference for them?
Unfortunately, we don't have very many observations, but when we did see them doing it in the field, it was on some sort of benthic substrate, so either rock or rock that was covered with little organisms. So yeah, I couldn't really tell you, unfortunately. I haven't just seen it that often.
Okay, because probably this can be a parameter for their geographic distribution, right?
There is another question for Paul from Byron Adams. We have found that things like distance from the coast and elevation integrate things like AC, soil moisture, etc. In your work, is it possible that physical predictors of biota also cover with things like elevation, distance to the coast, distance from ice, glaciers, streams, etc.?
Sure, of course. So we had elevation data as well, but I think might have not been enough. But of course, you saw that Mount Menzies. Oh, there was climate data in there. It wasn't relevant. There was everything that's location based in our analysis. So the Mount Menzies, Lake Terra Savoy, and Mawson Escarpment, of course, are location is proxy for all these things that you mentioned, and sometimes it comes out as relevant. So yeah, you're right. Hi guys, sorry, did you hear my answer?
Yes, we did. We heard
okay.
your answer.
Okay,
Thank you.
Thank you.
So, if there are no more questions, I think we can close the session. Fernanda, do you want to say something?
Oh yes, I want to close this block of our session because we will have an additional one. And first of all, I would like to thank you all for being here with us with the discussion, and also the authors who presented very, very interesting works. And I invite you all for participate of the next, I would say the last block of our session that will be at 7 p.m. UTC. So please calculate your time according to your time zone. When we will have additional presentations with more time for presentation. So we will have more time to discuss other topics in our session. So thank you all, and I hope to see you in around 2 hours. Okay. Eugenia, do you want to say something?
While we are waiting for Eugenia, I want also to remember you that there is also a poster session with very interesting contribution for this session. So I would also invite you to take a look to the poster and interact with the authors about their topics.
Okay. Eugenia, no words, Eugenia?
She wrote that she's not able to activate the microphone. So
okay, so that's it, guys. Thank you all for your participation, and we see you in 2 hours. Okay.
See you later.
Bye-bye.
Thank you. Bye.
Thank you.
Thank you. Nice to see you.
Okay, she said that she had some sort of reforming in the down floor, and she said, oh, it's impossible to say something. And also she said that she will not be able to moderate the session in the last block. So what do you prefer to do?
You can moderate the first half and I can moderate the second half. It is fine for you.
1, 2, 3, 4, 5, 6. Okay, 1, 2, 3. Okay, perfect. Romana, Christine, Louisa, and then you do with Byron, Anna, and Stephen. And we can do the opposite. So you open, I moderate the first part, and then you moderate the last 3, and then I close.
Okay, cool. Perfect. For stopping the recording, we have to do that, or the SCAR IT team can do that?
Hi, this is Manoj from tech team
Hi.
here. That was a great session.
Thank you.
Yeah,
Thank
you
you.
can pause the recording as well.
Okay.
Okay.